So this is the military database. So this is our chemistry building. And in general, most of structural biology and chemistry of NCI is located in Frederick, which creates a very productive, very useful environment for us, collaborative environment. My group belongs administratively to a newly formed Cancer Innovation Laboratory, which is unusual in the sense that it doesn't focus on any particular science field. So we're a bunch of different folks, immunologists, biologists, but the major spirit is collaboration, and that's how we function. So, and because of that, so our group has dual role and dual goals, I would say. So first, we generate chemical biology tools and drug candidates for molecular targets identified by NCI research groups. But also we develop novel effective methodologies and adopt novel methodologies in molecular discovery. And we're different from many other, I think, computational inclined drug discovery groups, that we not also do computational work, but as Mark mentioned, so we quite actively involved in evaluating the activity of our compounds using of modern
Is it any good? Yeah, it's quite good.
An array of biophysical, biochemical, and cell-based assay, which actually creates shortcuts. It's definitely a lot more labor intense, but same people do docking and other computational work, but also assays of the compound. But then, we can feed back the data that we generate back to the computational work. And although we are trying to delegate as much activity studies as possible to our collaborators, but most of them are biologists, so they would be glad, to do some mouse work, but not biophysical work. So that's why we do it ourselves. So the environment also such that we kind of, have to convince people around us in, terms that what we're doing is good and essential. And these are three most common misconceptions that we have to fight with. So the first one, you can't trust docking data. We can explore sufficiently large databases using experimental high throughput screens. And the scientific community has generated enough compounds for effective drug discovery, why to bother with virtual things? And in addition, we are working probably in one of the most challenging drug discovery fields. I have a second head at NCI. I'm frequently asked to give talks entitled, "Why haven't we cured cancer yet?" So, and one of the challenges I always discuss is that the targets that we are facing are mostly non-druggable, very challenging ones, because we want to target highly allosteric molecules that are signaling. So that the signaling mostly occurs through protein-protein interaction that are known to be very challenging for small molecules. And many germline mutations, causing particularly pediatric cancers, involve transcription factors having large intrinsically disordered proteins. But also the environment is such that we cannot say no for any target. So if a famous oncologist, a really good doctor, calls you and offers you to work on a really lousy structure, you cannot simply walk away from it. So that's why we take on lots of targets, otherwise people would, wouldn't work on because of lack of, good structural data and many other reasons. So that's why, my group has been focusing for many years on mostly peptide mimetics. However, we ran into a lot of problems with them in animals, and that's where, Mark and myself asked this question. So whether we can substitute those peptide mimetics in particular small molecules by expanding the searchable database. people, keep forgetting, and Mark mentioned that the chemical universe is not astronomically large. It's well beyond astronomical, because the estimate for chemical universe is 10 to the power of 63, while the number of stars in the universe 10 to the power 24. So it's much larger, and we're still looking at tiny fraction of that. So, and that's why, it would be very interesting to explore into those dark areas, because we don't know what's out there. So, and the answer to the question is yes, not just yes. I would say hell yes, because these are just examples of some successful projects that we had using ICM Pro. These are all targets on the list would be classic non-druggable targets, and I cannot fortunately mention many additional ones because they're novel targets discovered at NCI. So the reason, I think, for the success that we do docking really carefully. So although we're a chemistry group primarily, so we're trying to minimize chemistry, because not only it's laborious and time consuming, but also it's bad for the environment. So we'd rather, dock to make sure that the compound fits back. As a result, I think that our average success rate is pretty good. It's in the range of 30 to 50%. So about this percent of compounds, of tested compounds, are active. Of course, it depends on the cutoff for activity, but we've set our bar fairly high. We do not test compounds at concentrations above 10 micromolar. However, while doing this, and we have explored quite a number of targets, only currently we have 15 active screening campaigns. So, but we have came across this pattern that it's more all or nothing behavior. Some X-ray structures are close to 100% in producing active, whereas others had practically no actives at all. So this is one of the examples that we have been looking at. So this is famously non-druggable KRAS protein. It's G12D muted in the complex with a peptide inhibitor that was identified from phage display libraries. So it looks like a perfect structure from crystallographic point of view, 1.2 angstrom resolution. But none of the compounds that we have identified were active. So more so, we kind of, ran a secret comparison project here, because Megan Peach in Mark's group used Schrödinger's glide for the same purposes, and nobody won this competition because all compounds were inactive. Although, we had two other competitions, and ICM Pro was the clear winner. So, and again, that seemed to be a perfect druggable pocket. And although KRAS is a very challenging target, it was a good target, I think, for us to explore, because there is a wealth of structural data. So there are more than 300 structures of RAS proteins in PDB. And we explored many of them. Of course, many of them didn't have pockets at all, but some that were crystallized with complexes were suitable for that. And many of them were like this one, producing zero things. And that's again because RAS is highly allosteric, highly mobile protein. I think, crystallization probably does lots of harm to it. However, we were able to identify 3 structures that allow us to produce the inhibitors. So, and the thing that we learned from this is that it's worth to spend time working on the structure first. And that's why you have seen the version of this slide already. So this is why the very first step in all our projects is verification of crystal structure suitability for screens. Now we use mostly ChemSpaces library of on-stock compounds. It's 5.5 million, which we can easily purchase from them. Then we test the hits, and this is a point of go, no go. Although no go means that we need to work on the structure, not throw it away. So then we dock SAVI diversity set. So Mark recently created this 5 million diversity set, and this is a very important step. I think, any library that you are using for screening has three most important properties, and paraphrasing Julia Child, it's diversity, diversity, and diversity. So here we have a control of diversity. So then, we synthesize the hits from this whole thing, and in order to access as much as we can entire libraries, so we do fragment-based, we used to do fragment-based and 2D searches of entire database. And in addition to MuleSoft Glide, we also use Google's BigQuery through collaboration with Google and OntoCam. So, and that is pretty powerful search engine, but unfortunately, it's very expensive. So we use it for free because we collaborate on SAVI projects with them. So, but that was effective too. However, definitely 3D similarity searches are much more effective, and we had lots of success. And on top of this, they allow you scaffold hopping, which is a quite desirable thing in medicinal chemistry. So currently, we can search 5.2 billion compounds using RAID. But I'm happy to tell everybody that the new version of REAL, which is 6.6 billion, is almost ready. So we have generated conformers for that. So starting from next week, we could be accessing 10.6 billion compounds. So it was quite an undertaking for this new REAL version, although the latest modification that Eugene introduced to conformer generation made it a lot faster. It still took almost 3 months and over 40 million CPU hours. But since we're at an age we use your taxpayers' money on everything we do, we are happy to share all the databases, and so this is kind of commitment for the future. We will be doing that for the community. So then we redock, because docking still is the best way of assessing the compounds for the binding. But again, it's very rarely that you can fill the pocket entirely using database compounds. So that's why the last step is always using traditional medicinal chemistry, and we use computational tool, this tool that's present in ICM Pro Find group, so that, we find lots of use. But, the bottom line, so that we were able to identify compounds with binding in at least micromolar range for most of the targets, in even very challenging ones. So, and this is just, some real life challenges with protein structures, since, this is the title of my talk. That's what, I want to share some experiences with that. So, the most difficult one probably is when there is no structural data available at all, just AlphaFold model. And I was quite surprised actually when we got positive activity data in this particular case. So all we did is minimization of side chain in the pockets with ICM Pro. I don't think that it's gonna work for any case, but at least the very first try that we'd done was quite successful. So the other one, so the X-ray structure is available of poor resolution, and we do the same trick successfully, minimization of side chain in the pocket using ICM Pro. The case like with RAS, so there are several X-ray structure available, and we have this same thing with TIGIT, but protein is flexible. You can see that the structures are different, so and you don't know which ones to choose. So then we had no choice, since at the moment we can't predict which structure is going to be suitable, which not. We simply do it experimentally, testing VLS suitability by docking chem spaces library. And I should emphasize that we never order hell a lot of compounds, 5, 6. That's a typical thing that we test, and it works just fine. So the other example, when again, quite common thing, is that only cryo-EM structure available. It's a wonderful target, but it's humongous complex, and all you can get is poor resolution cryo-EM structure. So that allows, requires lots of work, but recently Hody Hoop in our group was successful using several available ligands that were identified by experimental high-throughput screening in order to optimize this fuzzy cryo-EM structure of the protein. She used flex docking of this ligand into the pocket, and then selected the conformation of the pocket that yielded the highest best score for the known ligand, and then used it in future screens, and she was able to find compounds that were significantly more potent than the original structure around which the protein was optimized. So we also had a situation when the target was an enzyme, and as many enzymes, it can exist in this closed conformation. Basically, there is no access to the active site that you want to target. And there is loop, because sometimes several loops that close up and down. So, and we're using molecular dynamics simulation. We just started using ICM Pro molecular dynamics simulation. So far we have been using the classical, CHAMP one, and it also seemed to work quite well. And also, very typical situation we face is that the pocket that we need to target is a really shallow one. And what we do is just expand database for docking, hoping to find something. And Mark already showed you the example of the STAT3 and terminal domains. So that had this very shallow pocket, and drug-like density was quite lousy, -0.5. But nevertheless, probably we were lucky, but it was successful. So the identified compounds are now in preclinical development. So, but in spite of all of that, so the challenges that we're facing are quite tough. As I mentioned, so none of the libraries that we use were able to fill the pocket completely. So, and in order to expand further, and we're kind of, looking into generation of a la carte libraries. So, many people use those fragment-based approaches for very good reason. However, we are also focusing very much so on the chemistry that is used. So, because our chemical space, our chemical galaxies are built around the transforms that we use in addition to the blocks. So, and for that, we need effective methods of predicting synthetic availability of compounds. And this is a very challenging thing that Ruben was asking about, right? So, because there are lots of currently approaches of predicting synthetic availability. I think, and I mean, it uses mostly the chemistry that they're comfortable with. And they have developed lots of nice chemistry that is targeted for high throughput synthesis, combinatorial chemistry. So that's what is different. However, they're trying to stay away from coupling reactions because they require expensive catalysts that they probably, and they are not as easy to parallelize. Right, so which we sort of, don't care because we need the compound, but for their, for their business model, it's important. So I already complained about computational resources. So, and we always will be short of those, and about the crystal structures that are impacted by the crystal packing. But as Albert Einstein said, that in the middle of difficulty lies opportunity, and there are definitely plenty of opportunities at the moment. And thanks for, to Microsoft, those opportunities kind of, really create an optimistic view for me. So the question that we're trying to look into is privileged transforms, privileged blocks, and also correlation of pocket properties with certain parts of chemical universe transforms. And this is something that we have tried to address recently, because everybody's focus is on the blocks.
It's 5 minutes left, Nadia.
Okay, thank you. Thank you. Yeah, so about done. So, and what that was definitely a very preliminary estimate for search for this correlation. So, but again, ICM Pro played a critical role. So we have taken Savi diversity set of 3 million compounds and docked it into 39 well-characterized protein pockets, looking for correlation with the properties of those pockets. And although we didn't intend to benchmark ICM Pro on these ones, but we had to evaluate the targets that we have selected for the suitability for VLS. So we redocked for all the structures that had their own ligands. We redocked those ligands and compared the poses for those ligands, experimental and predicted ones. And you can see that RMSDs were very impressive in all cancers. And these were randomly selected complexes. So they were below 1 angstrom in all cases by except 2, but in both cases, the ligands were sticking out into the solvent, and all the differences were actually outside the pocket. Otherwise, Microsoft did a fantastic job. And you can see that DLAD for those targets differed in quite range, and we tried to have both pockets suitable for small molecules and protein-protein interactions in these things, because this, as I mentioned, our big interest in the future. So, and DLAD were, not so good in many cases. And what we found, actually, drug-like density descriptor had a positive correlation with number of hits, but correlation was statistically insignificant. So what it suggests, so although it's a very useful parameter, but you shouldn't be discouraged by using the pockets that show not so good DLADs. And also it was surprising that the hit rates for 93 transforms used for Savi generation differed a lot. And clear winners were coupling reactions that form CC bonds. So they were, and you can see the differences were orders of magnitude in terms of productivity for different things. So the cross-coupling reactions that were particularly productive were Senegashira coupling, Suzuki Mayura cross-coupling, and Hiyama carbonylative cross-coupling. So that's, I think, a good message for medicinal chemists working in the future. Different transforms, different pocket properties have different impacts on the hit rates for different transforms. So this table is definitely impossible to read. But in the yellow, we highlight correlation coefficients that had that were statistically significant with p-values below 0.5. And the pocket that was the most essential and predictor of a significant number of hits was the number of HB donors. So the other properties that we looked at were all extracted using ICM-Pro Pocket Find. Buriedness, sphericity, aromaticity, hydrophobicity, area, radius, and volume. So you can read details and all in the manuscript that has been posted already, but I believe that this data can be very handy in the future for designing those slice-targeted libraries. And in conclusion, again, so we were very fortunate to have lots of terrific collaborators. These are our biology collaborators. So, lots of help from Ukrainian folks. And of course, I'm very grateful to Eugene and Andrew for all the help we're getting from them constantly. I can imagine that they're extremely